This is Srikar of BTech Biotechnology, Gokaraju Rangaraju Institute of Engineering and Technology. I did my project on recombinant DNA technology. This project deals with the cloning of STAT3 deletion mutants into pcDNA vector. So, what is recombinant DNA technology? Recombinant DNA technology or cloning technology is the procedure of isolating a defined DNA sequence and obtaining multiple copies of it in vitro. Cloning is frequently employed to amplify DNA fragments containing our genes of interest. So in this procedure of cloning a gene, we have used different equipment such as centrifuge, water bath, laminar airflow cabinet, ultra-low freezer, wild strains, enzymes, and bacterial cells. Coming to the general strategy of our DNA technology, we first identified the desired fragments, isolated their DNA, extracted a suitable vector for carrying this DNA, cut the DNA and the vector into fragments, joined the fragments, and then at last transformed them into the bacterial cells. Now comes our gene of interest, STAT3. STAT is an acronym of Signal Transduction and Activation of Transcription. The main purpose in cloning these genes is that they play a major role in immune tolerance and tumor surveillance. I have employed pcDNA 3.1 plus vector for this purpose. Choosing a vector is an important part since this should contain the required origin of replication, antibiotic markers, multiple cloning site, and the promoter regions. Here starts the procedure for plasmid isolation. I have isolated it through the columns since this is the most reliable method. It is a three-step procedure of binding, washing, and eluting through a nitrocellulose membrane, thereby giving out the highest concentration of the plasmid. Now, one of the important operations in cloning is the PCR amplification. Polymerase chain reaction is a technique in molecular biology to amplify a single or few copies of a piece of DNA across several orders of magnitude, generating thousands to millions of copies of a particular DNA sequence. We have added primers, dNTPs, DNA tag polymerase enzyme, ions, buffer solution for the reaction progress. The reaction goes on with the primer binding, elongation of the complementary sequence through tag polymerase, and separation of the strands. This cycle of primer annealing, elongation, and separation repeats, leading to the generation of thousands to millions of copies of the DNA. This is the gel picture I obtained after PCR amplification. The bands represent the DNA fragments at the respective sizes. The second part of the cloning is the restriction and digestion. Cutting the DNA at respective sequences with the help of restriction enzymes is called restriction digestion. I have employed ECOR5 and HIND3 restriction enzymes in this part. Both the DNA and the vector are cut with these enzymes, generating the same DNA sequences on both. This is the gel picture I got after running the gel with the digested samples. This picture synchronizes the PCR gel, indicating that the digestion is proper. Now comes the ligation. Ligation is joining the digested samples with the help of an enzyme T4-DNA ligase. The inserts generated by ECOR5 and HIND3 enzymes bind to their respective complementary sequences on the vectors, resulting in the formation of recombinant plasmid. Now the last part of the cloning, which is transformation and plating. Transformation is carried out in competent cells whose cell walls are loosened for easy intake of the foreign DNA. After transformation, they are plated onto the agar plates. We have observed the colonies after 12 to 16 hours of incubation. The clone thus obtained is declared positive by colony screening through PCR. I have isolated the plasmid DNA of these colonies and amplified them through PCR. I got a gel picture resembling the same that of the original PCR samples and the digested samples. Now I have the clone of the stand genes in pcDNA vector in the bacterial cells. This is how recombinant DNA technology works. I am very thankful to my HOD madam, principal sir and director sir for giving me this wonderful opportunity. Thank you.